Just forgive me, but I'm going to insist that anybody who is farther back than this,
It's never happened.
move up.
Come on.
Come on down. Come on down. Thank you. Don't be shy. As far as we know, no one has no business. He gets close to us. Even though he works with Michael. All right, I'm gonna start off with like the usual formal kind of intro, and then I'll throw in a little bit more personal stuff. To tell you about his work, I'm gonna read from the announcement. He's the winner of the International Society of Microbial Ecology Winograd Award of 2026. Award. He will give a plenary at their symposium, the meeting this summer. And I like the description of what he does. Noah Fierer, he probably wrote himself. Noah Fierer and his research group explored the microbial life found in a range of environments, including those bacteria and fungi living inside our homes, in soil, on plants, and in the atmosphere. The group uses various approaches, including DNA sequencing and high-throughput cultivation, to explore the diversity and structure of microbial communities, quantify biogeographical patterns, build a trait-based understanding of microbial life, and examine how microorganisms influence the health of ecosystems, plants, and animals, including food. And I'll just point out that he got that to start on the biogeography. I think, can I call you the father of microbial biogeography? And that goes back to your time as a postdoc when I met you. The papers came out in 2006, 2007, inside thousands of times. So Noah grew up in New York. Pennsylvania. He got his undergrad degree at Oberlin College, where for 1 year he was in a group of 4 that included Jan Aldenrad, who on Radiolab phase, and he was into the guy who started ZipRecruiter, and the 4th roommate was Gary Steingart, the famous novelist. So I guess, yeah, quite successful. He did his PhD at UC Santa Barbara with Josh Schimmel, then went on to do a postdoc with Rob Jackson and Duke, but he was still living in UC in Santa Barbara at the time. I went to Santa Barbara to do a postdoc, so that's where Noah and I met in 2004. As Sasha mentioned yesterday, Noah has crushed all the metrics that we use to quantify research output. To me, where he's most obviously a super outlier is citation. Go to his Google Scholar page, your eyes might pop out of your head. It's been cited almost 190,000 times. In 2025, his research was cited over 16,000 times. That's more than most researchers rack up in their entire career. Okay, so in 1 year, his work's been cited more than most of us cited in our whole career. And I think it's natural to ask, how can someone be that productive and do that much influential work? Maybe they're just a total workaholic. But this is where my personal insight comes in. Because, like I said, we met in 2003, 2004, share a lot of outdoor interests. We've been on trips together in the desert, mountains, trips. And I can tell you that on these trips, when there is an internet connection, Noah does often have his laptop open at night. But he's not working. He's like looking at maps and planning the route for the next day, or getting data on something. So I don't know. I cannot answer the question how if we do it. I guess he's just really good on it. So help me, welcome me.
It's far too generous. Can you hear me okay? Okay. So my instructions for this talk were to quote unquote have fun with it. That was all the instructions I was given. So we'll see. How many people here say like I'm a microbial ecologist? There's a few. Okay, this talk is probably not for you. No, just kidding. what I want to do is I want to give a talk like about microbial ecology for people that aren't microbial ecologists, right? And before I do that, well, one thing I want to say is like this is like all new, okay? So if there's things that don't make sense, if you have questions, if there's things you want to add, raise your hand, interrupt me, stop me, add to it, ask questions. So please do that. I also want to give warnings, 3 warnings. So what I'm going to talk about is this is a biased perspective on the field, right? Because we're all biased, right? So this is last night what I wanted to talk about, right? So you're going to get a bias. And I'll also say, like, I'll use a lot of examples from my group's work, not because they're the best examples, but because I was lazy and I had some of those slides and figures available. I'm like, all right, well, I'll use this as an example. Okay. It's going to be incomplete. I'm not going to summarize the whole field of microbial ecology, or for that matter, the field of ecology. And it's going to be an oversimplification. I'm going to make some oversimplifications about where the field of microbial ecology is, where it's been. I'm also going to make some oversimplifications about plant animal ecology. So please raise your hand and call me out, right? Okay, so before we get to this, let's start at the basics. What's a microbe? If you're a microbial ecologist, what does that mean you study? Like, what's a microbe? What's a definition of a microbe? We're starting at the ground level here. Come on, somebody help me out here.
Wrong, but organism that's smaller than can be seen with the naked eye.
That's it. You're not wrong. That's it, right? Okay, so when we're talking about microbe, I use that term all the time. Lots of people use it. It's basically not a super helpful term. It basically just means things that are small, right? So this includes viruses, includes bacteria, includes archaea, completely different phylum that's also microbial, includes yeast, like fungi, like yeast and so forth, filamentous fungi. It even includes fungi that produce visible fruiting bodies, like mushrooms. Like we call those microbes because they're kind of interacting with their environment in terms of the hyphae and the mycelia at a microscopic scale, but obviously we can see them, and some of them can be enormous, spanning hectares. You know, we're talking about diatoms, coccolithophores, oomycetes, amoebae, and other protists. We're talking about phytoplankton. The list goes on and on. So the only unifying thing is that they're small. So we're spanning like basically all the known diversity of life we just consider microbes. So many years ago, I hate to start with a quote that from one of my papers. If you like this quote, I wrote it. If you don't like it, blame my co-author, Jay Lennon. Maybe he wrote it. We don't remember who wrote this, but the term microbe is so vague as to be meaningless. As meaningless as if if biologists were to begin using the term macrobe to refer to all plants and animals. The main difference being that the human ego is sensitive to the idea of being lumped together with mosses and jellyfish. So I want to start out by saying that microbes, it's not a super useful term. So calling it a microbial ecologist, which I call myself, is also not super informative. So this talk has 2 parts. It's the classic bait and switch, right? You're familiar with the bait and switch, in like retail, like come on in for this great deal. Oh, we don't have that anymore. You get snapped, in the mousetrap, right? So I'm going to start with the bait. Why I think macrobial ecologists, so ecologists that primarily study plants and animals, should study microbial ecology. And then the second half is the switch, which is, what are some of the challenges and misconceptions in microbial ecology? So to phrase this another way, what I'm going to start with is like, what gets me excited about microbial ecology? The Sunday afternoon, I'm like feeling good. I'm like in a good mood because it's the weekend. What am I excited about microbial ecology? Then the second half is going to be that Monday afternoon after a hard day where I'm like, God damn it, like this field is frustrating. There's a lot of, so bear with me. So we'll start with the positive, the bait. Okay. What are some reasons why I think, from my bias perspective, microbial ecology is really cool and deserves to be better integrated into the broader field of ecology? One is just a really basic one. Most of the biological diversity on Earth is microbial, and I already alluded to this, but let's look at it from an evolutionary perspective or a phylogenetic perspective. Okay, so this is the tree of life as of February 2023. You can quibble about exactly how these branches are organized and so forth, but the key point is the plants and animals are a pistaconta, one group, right? So obviously, I'm not saying plants and animals aren't important, but in terms of the evolutionary diversity of life on Earth, we're looking at a fairly small subset. And the fungi are also in there. We have all these protists, we have archaea, we have bacteria, and so forth. So microbial ecologists are basically tasked with all of that other diversity that's not essentially plants and animals, right? And I think this is one thing that's really cool, and I'll emphasize in this talk is like, ecology is not that useful if it only applies to plants and animals and excludes all the rest of the diversity of life on Earth. That would be my argument. And what's not shown here? Viruses, right? Viruses don't have conserved genes, so it's hard to put them on the same phylogeny. But obviously, viruses are abundant in every environment, including in the air in this room right now, hopefully not pathogenic viruses. But, so if you look at, this is a classic photo from an image of like a milliliter of seawater, and you zoom in, you can see diatoms, you can see lots of bacteria, and all those really small green specks, those are bacteriophages, so viruses that infect bacteria. And they're usually 10 to 1 or even 100 to 1 viral particles per bacterial cell. So incredibly numerous. And there's this whole separate field of viral ecology. Like I think bacterial ecology is hard, fungal ecology is hard. Viral ecology is like really hard because they're just really challenging to study. We know they're important, or we think they're important, but they're just hard to study. And then just to emphasize sort of from an ecological perspective, what's some of the value of this diversity that we can play with? I just want to give you one example. Okay, this is from a study we did a few years back looking at soil microbial communities in the 50-hectare forest dynamics plot in Barro Colorado Island in Panama. Anyone familiar with this site? Familiar with the neutral theory? It comes from this site, right? It's actually one of the most boring tropical rainforest sites I've ever been to. It's like dead flat. But, so for context, we've got 288 tree species at this site across these 50 hectares. So we went and collected 625 soil cores from this plot. The study was really focused on matching the distributions of microbes in the soil to the plant distributions and so forth, but let's not talk about that. But for context, we have about 33,000 bacterial species. I don't want to argue about how we define a bacterial species because it is somewhat arbitrary, right? But and then fungal species, about 25,000 fungal species. So with this plot, so if you want to test theories, concepts, models, we just have a lot more diversity to play with here, even at the same site that, again, has been well studied by plant ecologists and theoretical ecologists for that matter. So just this enormous amounts of diversity we can work with. Yeah, again, feel free to interrupt. Another thing that I think has been really fun about microbial ecology, and really more just in the past few years, is that there's a lot of genomes for many bacterial and archaeal taxa are now available. So this is not just names of species or like single marker genes. We have whole genomes for a lot of these organisms. And that's incredibly powerful for some reasons I'll talk about. But just to emphasize the scale, so this is the most relatively, this was from 2025, so this number is now actually double in the most recent release. I just couldn't find a good figure. So currently there's, 700,000 bacterial genomes that are out there. And this is roughly about 200,000 different species of bacteria that we have genomes for. For archaea, we're about almost 18,000 different unique archaeal genomes that are out there. And in contrast, we have about 2,400 plant genomes. And this is not because people aren't trying to get plant genomes, it's because it's much harder, right? There's less diversity in the plant genomes are way bigger, right? So, and I will say that a lot of these genomes don't come from things that we grew in the laboratory and studied. This comes from, you just take a metagenome, which basically means DNA sequencing an environmental sample and assembling that genome. So that's where a lot of this growth in genomes comes from. So again, it's not the full scope of bacterial or archaeal diversity that we see out there in the world, but we now have a lot of genomes to play with, essentially, more so than plant and animal ecologists have. And one thing that's also cool about these genomes and these sort of databases that are available is that the taxonomy largely reflects phylogeny. For bacteria and archaea, for fungi, no, it doesn't. Protist definitely doesn't. And this is also true for a lot of plants and animals where like we have kingdom, phylum, class, order, family, genus, species that doesn't necessarily reflect their evolutionary relatedness. So one advantage of this is if you tell me the genus that a bacteria belongs to or the family or the class, it's telling me that all members of that group are from the same lineage. So what it does is it makes it easier to integrate sort of phylogenetic perspectives onto ecological perspectives, because we don't have to sort of redo the tree. They've been integrated. So the taxonomy and the phylogeny are by and large coherent. And I'll give you an example of why that's advantageous. So why not fungi? I think it's because the study of mycology or the study of fungi is just older. So it's been harder to sort of, this takes a lot of work because you have to convince everybody to change the names of things. You're like, you used to call this two different families. It turns out they're actually evolutionarily related. So let's lump them together. And then likewise for splitting. And for most microbial ecologists, they're comfortable with changing the names so that it aligns with phylogeny. Plants and animals, that's harder to do because they have this long history of taxonomy that no one's just willing to like overwrite whole hog and sort of make it line up better with phylogeny. Okay, so we have all these genomes now, and we have a taxonomy that largely aligns with phylogeny. So we can do things like this. So this is an example of a paper that just came out like a week ago. We set out with sort of this idea: can we construct a periodic table of bacteria to map bacterial diversity in trait space? So, everyone's familiar with the periodic table of the elements, and it's really useful for understanding elements, how elements interact with one another, and the properties of elements and so forth. And one thing that's really frustrating about bacterial diversity or any microbial diversity is just the amount of diversity. So how do we start sort of collapsing that diversity and understand like groups, functional groupings, or at least groups that share similar traits? So this is just our first sort of proof of concept on how you can do this, leveraging all these genomes and leveraging the phylogeny that we have. So I won't go through all the details, but for this, we just looked at traits we could infer from genomes, which is a limited number of traits, like oxygen tolerance, like phototrophy. Can it use light to generate energy? Autotrophy, is it capable of fixing its own carbon, and so forth? Genome size is in there. And because we have this coherent phylogeny, we can say, okay, where in the phylogeny do we see breaks where all members of that group share similar traits based on just this small subset of 6 traits? So then that becomes a cell where you say, okay, this is a functional grouping that share similar values and trait space that we couldn't do if we didn't have that phylogeny that again matches up with the taxonomy. So obviously I won't go into it, but you can look at this, you can say, okay, here's what members of this taxa go in there. And so the idea is that we can start sort of interpreting the bacterial diversity we see out in the world, and someone could say, all right, we know something about members of this group, and some of these traits are conserved at phylum or class level, and some are only conserved at the genus level. But you can sort of scan across a phylogeny to see sort of trait cohesiveness. Okay. The other thing that I think is really cool about microbial ecology is we can directly integrate evolutionary processes alongside ecological processes, because they can evolve at time scales that we can do ecological work, which you can do with plants and animals. It's just much harder because they just evolve slower. So the time scales are just different, right? So there's a great example of this. I won't go into the details, but it's a super cool paper where this group had studied Lake Mendota, which is right next to Madison at University of Wisconsin. And they had really beautiful time series data over the past 20 years. And again, I recommend reading this paper so they can describe ecological patterns, which taxa sort of how they change in their abundances, seasonally and over time. But they can also look at sort of evolution, and what they show is that for certain groups, you can see sort of accumulation of mutations and selection for particular traits during like the summer versus the winter. And then it'll reverse back. So not just assortion, not just selection at the sort of species level, but actually changes in the sort of the genomic and phenotypic traits of these individual strains, which is pretty cool. Okay. One thing, so a lot of people in the ecology center are sort of more on the ecosystem ecology and biogeochemistry side. And so one question I get all the time is like, okay, I've got all this microbial data. What does it tell me what this soil is doing, or what this stream is doing, or how carbon is cycling and how nitrogen is cycling? And the answer is often like, I don't know. Like it's hard, right? It's hard to interpret for some of the reasons I'll get into later. But one thing I think is really useful about this microbial data, whether it's bacteria or fungi, is we can use these microbes because they're so diverse, because they're so abundant in basically every system, we can use them as bioindicators. And of course, we can use plants and animals for bioindicators of pollution and so forth. But microbes, just because the breadth of diversity, there's a lot of power there. And not just we can use taxa as bioindicators, but also use specific microbial genes as bioindicators to measure environmental variables that we think are important, but are hard to measure directly. So use the microbes to tell us the conditions in that system. So let me just give you two examples, again, from projects that I've been involved with. One is, if you study plant ecology, phosphorus is often really important. Measuring phosphorus availability is really hard because measuring how much phosphorus is actually available to plant roots is tricky. You have to do all these chemical extractions, and then you're making all these assumptions and so forth. So what we did with this study is like, well, let the microbes tell us phosphorus availability. So there's a lot more to this, but I'll just say we identified a subset of genes that are really bacterial genes that are really predictive, that are highly correlated with phosphorus availability across hundreds of soils. That was then tested with an independent data set of hundreds of other soils from a different continent, and it works pretty well. So, what this sort of provides is like, here's sort of a more rapid way to assess phosphorus availability without doing the complicated chemical extractions and so forth. Another example, simple one. In a lot of environments, aquatic environments, sediments, soils, you name it, human guts, oxygen is really important for determining what organisms can live there. Oxygen is really hard to measure. So we can look at ratios of genes from bacterial genes to say, what's the percent of aerobes, bacteria that require oxygen, versus anaerobes, bacteria that don't require the presence of oxygen. And we can use that to infer oxygen levels in the environment. And this is an example from 9 infant guts. Good luck trying to measure oxygen levels inside the large intestine of infants. You can do it, but it's not trivial and very challenging. So you can start looking at changes in oxygen as they go from birth to up to 900 plus days. That tracking, and again, environmental variable that's just hard to measure directly. And this was validated with all sorts of different ways that I won't go into, but again, just an example of using microbes as bioindicators. Okay, we're still on the positive part here. We haven't gotten to the issues yet. That's coming. I think the other thing that I find personally interesting about microbial ecology is there are all sorts of opportunities to study microbial ecology in engineered systems, right? Like hot water heaters, which is my personal obsession the past few months. It's a really cool system, right? It's bioreactors, fluctuating water flow through, hot, of course. They also are loaded with a lot of hydrogen that's actually generated by abiotic, like the sacrificial anode that you have to prevent rust. So there's all sorts of cool things that you can do with hot water heaters. You can have replicates and so forth, alter flow rates, change temperature, change nutrient supply, so forth. Shower heads, we've done a lot of work, not with shower heads per se, but also looking at plumbing biofilms and looking at succession in these environments and the biogeography of pathogens in these systems, looking at homes and house dust and what microbes are living around us and how that's shaped by having a dog or cat, which is the #1 most important factor determining what types of bacteria are found in your home, is whether you have a pet in your home. Looking at sewage communities, a lot of work on this and understanding how to treat sewage better. So there's just lots of opportunities. But my favorite example of an engineered system where we do microbial ecology is actually sourdough. And in my defense, we started this project before COVID, before everyone got into sourdough. It came out right after, but it was started way. And and it was just a super fun citizen science project because people, it turns out, I didn't really realize this, people are obsessed with their sourdough starters. And we would get these submissions of people sending in their sourdough starters that came with 3 pages of handwritten notes about the history of their starter, how their great-great-grandmother during the Yukon Gold Rush, extracted this from their husky, or who knows what. But you can start looking across the diversity of sourdough starters, whether there's a biogeography to it, whether there's associations between certain bacteria and certain types of yeast that we always tend to see in these starters, and so forth. And the other thing you can do is you can do a common garden experiment, a classic sort of ecological experiment. You can take all these different starters and put them in the same substrate. So same flour, same amount of water, same temperature, and all these replicated vials. And then you can see, does it matter if you have different starter communities. When you control everything else, does it matter for the flavor of the sourdough? Does it matter for how quickly it rises? You can measure all these different compounds and start relating the types of microbes that you find in the sourdough starter to like essentially the flavor chemistry and the aroma of the sourdough. We even had a panel of people that were like expert tasters. A lot of this, or sorry, expert smellers, I should say. So a lot of this was done with just the dough as it was rising, not the final product. And I was like, this is never going to work. Like these expert smellers, like whatever. And their data, like in terms of their like comments on vinegariness and all these things, like directly aligned with the volatile analyses off the GCMS. So I was like, okay, maybe these people actually are expert smellers. But it was amazing. Like we would get randomized, and give them replicates, and they're like, this is exactly like sample number 23 that I smelled. So it was pretty cool. But just an example of the kind of sort of ecological work that you can do in engineered systems. Another reason, you can do experiments with microbial communities that are difficult, not impossible, but difficult to do with plant and animal or animal communities. Example, succession, right? Chapter 2 of the intro ecology textbook is on succession patterns, maybe not chapter 2, but early on in ecological textbooks. It's like sort of a canonical study, look at how, for example, plant communities change over time. And when you're studying plant communities, you often have to use a space for time substitution because it's hard, not impossible, but it's hard to look at succession in real time because plants take a little while to grow, right? So like, for example, people look at glacial recession and look as you move closer to the glacier, as you go to like younger systems from older systems, how the plant communities change. Everyone's probably familiar with this, right? But the nice thing about microbial ecology is you can track successional dynamics in real time. And you can do replicated successional experiments, right? Because they just grow fast enough. So one example, again, another food example. If I were to do this all over, I'd probably become like a food microbiologist, but I'm not. So we just do these side projects. So this was actually a graduate class that I taught. It was focused on metagenomic sequencing and bioinformatics, but we actually designed an experiment working with brewers at Colorado State, looking at wild fermented beers. So who's familiar with wild fermented beers? A few people. So these are beers where you don't directly inoculate a yeast or bacteria for that matter. You just leave it open. You leave the wort open and like whatever's blowing around just lands on there and starts the fermentation process. So, we did this experiment like if it's outside versus inside the brewery, does you get different? And then tracking over time, is there hops there or not hops? And basically looking at successional dynamics in these communities, because they usually start with a yeast. Then they become more diverse. You start getting all these different bacteria, different types of bacteria as time goes on. And how repeatable is this? Is there a priority effect where the first thing in there sort of dictates the successional trajectory and so forth? So just one example of many of ways that you can do sort of succession type studies in real time with microbial environments, microbial communities. So questions? I'm blabbing on here, so feel free to, if there's questions or comments, feel free to say so. Otherwise, I'll just keep going. Yeah. Oh, I shouldn't have put this up if I wasn't prepared to. This hasn't been published yet. It's hard to get 25 graduate students together to co-author a paper, but I think it was some sort of control group. I don't remember that, sorry. But the treatments were hops, no hops, time, of course, wasn't really treatment, but time, and then indoor versus outdoor, whether it was exposed to outdoor inoculation versus in a brewery. And there was all inoculated by yeast, but very different yeast. So indoor, not surprisingly, it's mostly commercial yeast because they're just blowing around in the room. And outdoor, we get same Saccharomyces, the same genus, but very different strains. And we think a lot of these actually come from like wasps because they were really attracted to the smell of the wort. And we did sort of anecdotally sample some of the wasps and they were like loaded with Saccharomyces, even if they had never touched the fermenting wort. So, and there is some evidence for that for like wasps and like vineyards where like they'll actually move the yeast around between different vineyards. But yeah, other questions? Yeah.
I'll go paper, but maybe you can tell me. So if I brought starter from the Czech Republic to the US 4 years ago, and my sister still has it, is it likely that it still has a signature of that origin, or do they just homogenize? We've always wondered that.
Yeah, so one,
Like, introduction,
uh,
is it just where they
to some degree, like we collected as much data as we could from people on like where their sourdough starter came from. And sometimes it does, I mean, it also matters like how you replenish it, like what type of flour you use and so forth. So they will change over time and like how it's stored, you know, whether it's at room temperature in the fridge and stuff like that. But we set out because everyone's like, oh, the San Francisco sourdough is like super, no. We don't actually see strong biogeographical patterns. And a lot of it is because like the commercial yeasts were actually the most abundant in almost all the sourdough starters. And I think it's just because if you're doing any baking, those yeasts are around. I mean, this is my guess that your sourdough starter may have started with some unique Czech Republic yeast, but eventually it sort of gets overtaken by sort of the high yielding commercial ones. And that's not always true, but oftentimes that's true. So yeah, sometimes it's a little disappointing because people thought their starter was so unique and then they see it compared to all the other ones. They're like, oh, my community is not that different, but there is a lot of diversity across the starters. Some of them, you know, they have a lot more like acetic acid bacteria, and you can see that in the rising dough. They see a lot more sort of like vinegary volatiles and so forth. So, okay. The other thing is there's a lot of opportunities to test ecological and biogeographical theories outside plants and animals. So this may be a little bit controversial, but I would say if your ecological theory or your biogeographical theory only works for plants and animals, it's not very useful. Full stop, right? If it only applies to that sliver of biological diversity on Earth, maybe it's not like a universal theory or a universal pattern. So one example, well, first of all, I would say as ecologists, we don't actually have that many theories that we claim are broadly applicable, but that's a whole nother discussion. I'll let Peter lead that one. But one of them, and I won't say this is universal, but it's commonly observed. Most of you, this is probably chapter 4 in the ecology textbook, latitudinal gradient and diversity, right? So this is just an example showing mammals. Birds show more or less the same pattern. Diversity increases as you move towards the equator. Not true for all plants and animals, but many groups of plants and animals, we see this pattern. And of course, much ink has been spilled trying to explain why this is the case. So many years ago, we said, all right, is this true for bacteria? And the answer is no. Okay, and this has been followed up more recently, and people still say the answer is no, right? So if you look at mean annual temperature, or first off, if you look at latitude, no correlation with, and this is soil bacteria, no correlation with latitude. Mean annual temperature, also not correlated with diversity. Potential evapotranspiration, things that we know for many groups of plants and animals are often associated with diversity patterns at these large spatial scales. Doesn't work. Instead, for soil bacteria, it's soil pH. That's what matters. It's the pH. And this is published a long time ago, but lots of people observed this more recently. And in fact, it's such a strong effect that if you want to look for other patterns, you first kind of have to control for pH, because otherwise it just drives me nuts. It just overwhelms the signal, this pH difference. And you're probably saying, oh, this is just bacteria. What about fungi? Okay, fungi, this is a paper that recently came out. They looked at, just look at the top here. This is sort of latitude away from the equator. And again, they expected diversity to drop as you move away from the equator. And not really, right? It's basically flat. Yeah, you see deserts have lower fungal diversity, really arid systems, but there's not this strong latitudinal diversity gradient that we observe for many groups of plants and animals. They would actually argue that this is evidence for a latitudinal diversity gradient, but I mean, look at the data, right? If it's there, it's not very strong, right? So again, it may apply. Latitudinal gradients often are observed for plants and animals, but oftentimes not for bacteria. There's also great examples for marine bacteria where they often don't see it either when they sample from the poles down to the equatorial ocean. So yeah. And then my last point on the sort of positive part before we get to the challenges is I would make a plea for, if you're an ecosystem ecologist, population ecologist, community ecologist, I think the microbiome and sciences in general needs more ecologists, needs more people with like sort of an ecological approach to thinking about the world. And I'll give you one example of this. Okay, how many people have heard of Clostridioides difficile infections, C. diff infections? They're really nasty. If you get this, it's bad. It's very hard to treat, often very persistent, can be life-threatening. And it's usually caused, and it is caused by a bacterial pathogen, Clostridioides difficile, that's very rare in communities. So if you look at this stacked bar chart showing all these are human gut samples. Clostridioides difficile does not show up on here because it's usually like 0.2% of the community. It's very rare. So it's not an abundant community member, but these are all healthy patients. So the different colors just represents different taxa of bacteria in the gut. These are all patients that are infected with Clostridioides difficile. You can see it doesn't take statistics to tell you that these communities are very different. If you have this pathogen in your gut community goes in a whole different direction. You get very different taxa emerging. This is classic ecology, right? And then they even do some ecological analyses. Ordination, right? You all do that in community ecology, diversity analyses, Shannon index, species counts, right? All this stuff. And I use this example because this is just an example of how, this is essentially ecological data, like what's causing this state change in the communities and what does that mean? And this is like an example of an invasive plant coming in, and then you get totally different plant communities over time developing. So lots of corollaries. And I also bring this up because I work with a lot of microbiome scientists, and they typically come from more cell and molecular biology backgrounds. No criticism of cell and molecular biology, but I was on a committee meeting once, and this molecular biologist says to me, it'd be really useful if we had a method to quantify how different two communities are. And I was like, do you know there's a whole department that's like 50 yards away where they've been doing this since like 1910, right? But anyways, and the other example is just, sort of where I think an ecological understanding, or at least the ability to handle messy ecological data, which all of you are experts in. Examples like probiotics. Like you go to any grocery store and you can, like, particularly if you go to Whole Foods, there's like a whole aisle of probiotics, right? This is an ecological question. Like, how do you introduce a beneficial bacteria and have that bacteria sort of change your gut community? Will it even survive in that community? Will it colonize, or will it just get kicked out? How do you identify those beneficial organisms? How do you modify your gut microbial communities? Or likewise, the similar sort of probiotic idea for plants, right? We know that if you add or remove certain microbes, you can make plants grow better, whether it's bacteria, fungi, or maybe even viruses. But most of these studies are done in sterile systems. So it's pretty easy to modify the community. But once you have an intact community like we have in the real world, it gets much harder to figure out how to modify those communities. Again, sort of broadly, it's ecological questions and ecological challenges. All right, we ready for the bad news? The switch is coming. But first, any questions about that? Okay, and there's probably lots of other reasons you could come up with, like essentially why microbial ecology is cool. If I had thought harder, I could probably come up with more reasons, but I think that's enough for now. So what are some of the challenges and misconceptions in microbial ecology? What annoys me about the field, or frustrates me about the field is probably a better way to say it. One, and I already alluded to this yesterday in the talk, is most microbial taxa remain uncharacterized, or at least poorly characterized. This is true regardless of the environment you're looking at, even the human gut. We have a lot of genomes for like nearly all human gut bacteria, more common human gut bacteria, but that doesn't necessarily mean we know their traits, right? And one way this is often summarized is, we'll do a lot of sequencing work on a soil sample or a sediment sample or a river sample, or it doesn't matter. And there's some microbe that appears to respond to some perturbation or treatment or whatever. And someone will ask me like, well, what does this microbe do? And I'm like, well, first off, like, I don't think that's phrased the right way. If I tell you there's a plant species here, no one's going to say, well, what does that plant species do, right? And it's too broad a question. But the most frustrating thing is oftentimes I can't really answer it. I'm like, I don't know. It's a member of this group. Other members of this group tend to have these characteristics, but I'm not really sure. So it's a hard question to answer. And I showed this slide yesterday, so I won't belabor the point. But just to emphasize the scale of the problem, so even if we look at the most ubiquitous, most abundant bacteria in soil from like soils across the globe, we often have isolates for less than 10% of them. And most of the taxa we see, they're not closely related to anything for which we have genomes already available. So there's just this huge amount of uncharacterized diversity that's out there. Another example, So a few years back, I worked with a group that was trying to compile all the phenotypic trait data we have for bacteria and archaea that's published. And this is phenotypic trait data that comes from growing bacteria on a Petri plate and saying, is it pigmented? Yes or no? Does it require oxygen to grow? Yes or no? What temperature does it grow best under? Sort of fairly standard traits that are easy to measure. And one thing that was depressing for me, and I'm not even showing it here, is just the phylogenetic breadth was really limited. It's basically we had data for like 2.5 phyla. And then all the other hundreds of phyla that we see, we just have no data, largely because not only have they not been grown in the laboratory, they haven't been studied and done these sort of trait-based analyses. But just as an example, doubling time. This is doubling time under ideal conditions in the lab. We have data for I think in total it was like 800 different bacteria, which is again, a minuscule fraction. And then of the fraction of the diversity, it's even lower. Optimum temperature. So we have data for lots of bacteria on their optimal temperature for growth in the laboratory. But if you look at this data, I'm not showing it here because I didn't have a good example of this. 50% of the data points are at 37 degrees C. Is that because 50% of the bacteria on Earth grow best at 37 degrees C, or because most incubators in the labs are set at 37 degrees C? It's probably that, and also most people are concerned with what will grow at human body temperature. So even really basic traits like this, we don't have good data on that. And this is not to mention, the whole slew of other traits we would like to have and characteristics we'd like to know about microorganisms. And I already said, started out with oh, but look at all these genomes we have, right? So this is great. I think it's important to mention, I sort of hinted at this yesterday, that a lot of relevant bacterial traits and environmental preferences are really challenging to infer from genomic data currently. And so for example, substrate preferences. Does that organism grow better on amino acids versus ammonia? It usually has transporters for both, but we can't tell substrate preference. What temperature does it grow best at? Currently really hard to predict from genomes. The list goes on. A lot of traits that we need for inferring sort of ecological dynamics are very challenging to infer from genomes. Even things like salinity preferences, desiccation tolerances. We currently can't predict that from genomic data. And there's many reasons for this, right? Genes doesn't necessarily equal traits, right? There's lots of reasons. But one reason is we're horrible at annotating genes. So this is an example from E. coli. Of all the organisms on the planet, this genome is by far the best studied. Right? It's been, I think the first sequence for E. coli genome was in 80s, late 70s, something like that. And people have been annotating genes in E. coli for decades. So this is a recent paper. They said, how many genes that E. coli has have been characterized? So well-characterized is about 54%. Uncharacterized is 16%. Right? So these are genes that we know are in E. coli, but we have no idea what they do. So you can see the challenge here. And this is E. coli, right? You start looking at other bacteria, or for God's sake, you look at fungi, it's even worse, right? There's so many genes that we can pick them up. We can do all the bioinformatics we want, but we don't know what that gene is. So inferring traits from genomes is really hard just because that first step of annotating those genes is really limiting. So for bacteria, all of our annotations are based on E. coli, sometimes Pseudomonas, and a few other model species. And we're extending that to many, many different phyla that have not really been studied in the lab and those annotations verified or validated. So we use genomics all the time, metagenomics all the time, but we have to be really careful. You can generate a hypothesis, but it won't tell you what that organism is doing. And oftentimes we don't know when it's failing and when it's working. Okay, simple one. Most of you that have ever looked at microbial data, even in a paper, you're probably familiar with this one. This is a pretty basic one, is that we're really good at generating relative abundance data. We're pretty bad at generating absolute abundance data. So if you've read through microbial ecology papers, you probably got really annoyed. I get annoyed, right? And I've contributed to this, so I'm not blaming anyone else, right? You see the classic stacked bar charts where they all sum to 100%. So you can look at relative abundances of taxa or relative abundances of different genes in a community, right? Looking at visualized heat maps, or worse comes to worse, you see the dreaded pie chart, right? Where we're not looking at changes in absolute abundances of genes or taxa, just relative abundances. So I want to give you an example, and I think this is probably obvious why this is limiting, but I just want to give you an example. Okay, so recently This is not published. We just did a study looking at fungal allergens in the air in Denver. So this is one site in Denver. I'm just showing data from one site. So these are all the known fungal allergens. These are fungi that you breathe in can trigger the dreaded seasonal allergies, right? Runny nose, headache, red eyes. You all know the symptoms, or probably 35% of you know the symptoms of this, often triggered by molds as well as pollens, but here we're just looking at molds. So if you look at this as relative abundance, this is a time series from June until October, right? It's basically 3 fungal genera are most abundant. So you look at this and be like, well, it really doesn't matter what time of year I'm outside. My likelihood of developing allergies from these mold or fungal exposures is probably irrelevant. Okay, now let's look at the same data where we incorporate data on the amount of fungal spores found in the air. It's a totally different story, right? If you're outside going for a jog or a hike on this day, you're way more likely to get sneezing, runny nose than, in this day, even though the taxa are the same. And again, it's just simply because there's just way more of them out there. So there's lots of methodological reasons why our data is often relative abundances, but it can obscure some really fundamental patterns because we just don't know how absolute abundances of these taxa, or the genes for that matter, are changing across communities or gradients and so forth. Another one, detection of DNA, and this is true whether you're looking at streams or sediments or soil, you name it, does not necessarily mean that microbe is active or even alive. DNA is a remarkably stable compound, and DNA is not the only method, only approach, DNA-based approaches are not the only ones we use to study microbial communities. But if you judge the literature, it's like 95% of microbial ecology. And we all know this, and we kind of avoid thinking about it. So as an example, in soil, about 40% of the bacterial and fungal DNA is so-called relic DNA. What does relic DNA mean? This means DNA that's not from an intact cell. It's extracellular DNA. It's DNA that's out there in the environment, or at least in a dead cell. And this could have big influences on what taxa we think are alive in a given sample over time. It can also affect our estimates of diversity, because a lot of that diversity tends to go down pretty dramatically when you remove that relic DNA and just look at the DNA associated with intact cells. So there's 2 ways to view this. One way is it's a problem, and the other way is this provides sort of a more time-integrated perspective on communities, because at one point it was there. But how long that relic DNA, the DNA that's outside cells, persists in soil or other environments? Don't know. Minutes? Probably not. Probably longer than that. But I don't know the answer to that question. Is it decades? We know from ancient DNA work that it could persist for millennia, right? But we don't know. So we don't know what time scale our DNA-based approaches are capturing in terms of, like, for example, community dynamics. And this is like a longer list of annoyances than I thought I had. So bear with me. We're almost done here. Okay, co-occurrence does not equal interactions. Okay, so microbial ecologists, what we often have historically done is we look at the plant ecology literature for cool stuff, right? So for example, read plant or animal ecology literature and be like, look, we can construct these cool networks that show pollinator interactions, like which moths and butterflies and bees are visiting different flowers and different plant species and so forth. And we can put that into a network to like visualize those interactions. Or, classic food webs. We could say which taxa are eating other taxa or being eaten by, and we can assemble this into a network to capture food webs and food web dynamics and how that changes across space and time and so forth. So microbial ecologists say this, like, let's build our own networks, right? And I'm using examples from my own work because that way I can criticize it, right? We do the same thing. We generate these networks. So this on the left, don't worry about the details. This is like bacteria that associate with different species of moss. These are primarily nitrogen-fixing bacteria that associate with different species of moss. Here, this is a study in the UK where we looked at different bacteria, protists, soil fauna, and fungi that associate with different grass species. And we can start. But this is just co-occurrence. This is just correlations. It's saying when we see more of this thing, we also see more of this other thing. So when we see these moss species, we also tend to see these bacterial. This does not mean they're interacting. Like, you won't see predator-prey dynamics in these types of data, because almost by definition, they're not, very well correlated, right? But so it's co-occurrence. So in plant animal communities, oftentimes, I know it's challenging, but we can directly observe interactions. In microbial world, it's really challenging. So we assume co-occurrence equals interaction, but all it means is that tended to be most abundant at the same times or at the same space. Saving the most obvious ones for last, microbes live in microenvironments. Okay, if you want to study the environment that a tree is living in, we can do a pretty good job of like, all right, here's the light exposure, here's the temperature, here's the soil moisture conditions. And I know it's not that trivial, but we can do a pretty good job. If you want to know what environment a bacteria is living in soil or in sediment, or for that matter, the human gut, it's really hard. So just one example, bacteria, for example, live in biofilms. So the environment for a bacteria living inside this biofilm, these are these like, if you want to know what biofilm is, take your fingernail and scrape off the stuff from your teeth. That's literally a bacterial biofilm, especially if you haven't brushed your teeth in a little while. It's these like mucousy, and then bacteria do this for many reasons, but the environment inside the biofilm is going to be very different than from like a planktonic microbe that's living outside the biofilm. They can exchange metabolites with other bacteria. The oxygen concentrations are going to be different. Nutrient concentrations are going to be different. The list goes on. And we can't capture this with most of our analyses. So we don't know the environment that bacteria is actually living in. The pH can be dramatically different, right? The pH inside these biofilms can be like pH 4 versus outside the biofilm can be like pH 8. And that's really hard to capture that. Last point is a scale of sampling for microbial ecology is often far, far larger than the size of the organism. Okay, you don't believe me, so let me give you an example. Let's say you're a plant ecologist with a lot of energy and your plots are 1 hectare in size, a pretty large plot. And let's say the footprint of a tree is 0.2 of a meter squared. Don't quibble with the numbers, but it doesn't matter, right? So that's the footprint of that tree in that plot, not the canopy size, just the footprint. Okay, so now we're dealing with a scale where the individual is 50,000 times smaller than the plot size. Okay, now let's sample my favorite environment, soil. Let's say we sample 1 cubic centimeter of soil, which is roughly what we'll often sample, like just for argument's sake. And we're studying bacteria. Let's just say their average size is 1 cubic meter. So what's this disparity between the scale that we're sampling and the scale of the organism we're trying to study? 1 trillion. So we take samples that make sense to us, but from a microbial perspective, it doesn't make sense. So the equivalent is you're studying plant communities across all of California, and you're just lumping it into 1 sample. So like, yeah, look at all this diversity. Yeah, it's because there's like an enormous amount of environmental heterogeneity that you're capturing in there. And you're not necessarily capturing a community as we would traditionally combine. You're capturing lots of different communities in these environments. So we're often not sampling at a scale that's actually relevant to the scale at which bacteria or other microbes are experiencing the environment and the scale at which they're interacting with other organisms. And this is not a criticism of the field. It's hard to do, right? It's hard to do studies at sort of micron scale, or even for that matter, millimeter scales, right? Okay. What I want to finish with is just 2 slides, to try to bring all these fields together. And I think regardless of what type of ecology you study, this is, from arguably one of the first textbooks in ecology. And most of you are probably familiar with this quote. I think it's still very applicable today. Ecology is a new name for a very old subject. It simply means scientific natural history. And sometimes this is used in like a derogatory way. They're like, oh, you're just like counting birds out there, right? But I don't think it should be considered that, right? And like, I think the reason I mention this is a lot of what we know about plant and animal ecology is rooted in natural history. We know an enormous amount about plant and animal natural history. And we can leverage that to design really good ecological experiments, to interpret our data and so forth. And it's much harder for microbial ecology. So it's not that, we're sort of still at that natural history phase. And it's hard for us, it's challenging. It'll take some time in most systems. I mean, there are some exceptions, like some of the food systems where we do actually know a lot more about the individual taxa, but we're kind of stuck in this discovery phase. But I do think one place where plant animal ecology, ecosystem ecology, microbial ecology, I think what we're all struggling to do in the field is move from description to prediction. We're all really good or reasonably good at description. You know, like just something like we can describe what happens in a landscape with permafrost thaw, or post fire, or drought responses. And I'm not saying that's easy, but we have the tools to describe the responses of communities and populations and ecosystems, land use change, the list goes on. Where we're not good at is predicting those changes a priori. This is true for microbial ecology, plant ecology, animal ecology. And the reason we're not good at it is not because we're dumb. It's because it's really hard. This isn't physics. Physics is easy, right? This is hard to do, right? There's this context dependency. There's all this diversity we're working with. There's all these things to consider. And that's like, I think one of the big challenges in ecology is we oftentimes, not always, but we often want to move into prediction, and it's hard. So this is where I think, just to finalize, this is just one place where I think we do share a common sort of desire oftentimes is sort of move the field to this prediction and develop sort of predictive models that apply in ecology, regardless of the system. So that's it. Hopefully we have time for questions and so forth. So I don't know if we have time. So sorry, I got over enthusiastic. So yeah.
So awesome talk. I've noticed the problems that I like to focus on scientifically tend to be really messy, or I have like lots of data, really confounding factors. Like, great example, like I'm a genomics person, right? But I've also get to do community ecology. I also have this interest in, I think, microbial communities kind of for the same problem, unfortunately. And I kind of wonder, maybe if you're a good person to ask, how to go about making sense of problems that are like massive data, linear relationships, confounding variables. Like, when you're faced with that kind of problem field, how do you start?
I mean, that's a good question, and I'm not going to answer that. I don't know. But I will say you're probably not unique in this room of having to deal with messy data that's hard to interpret and so forth. That's ecology in a nutshell. So one way you could phrase it is that makes ecology frustrating because we're never getting like perfect correlations, right? And if you are, there's probably something wrong, right? But the other way to view it is that ecologists actually have an amazing skill set to deal with messy data and try to figure out what's going on, because the real world is messy. Like, you're dealing with economic data, it's messy, right? So I think ecologists were broadly trained, whether you know it or not, whether you like it or not, to deal with messiness. And the world is messy. So I think that's a really valuable thing, that if you're starting your PhD, if you're finishing your PhD, you're getting trained in the messy reality, right? So I didn't answer your question, but I don't know the answer. That's why. Yeah.
So, you're our field ecologist. I thought your point about using microbes to infer things that are difficult to otherwise measure is a really, really powerful idea. Where would you start if you're a field scientist? Where would you start looking for how to incorporate that kind of analysis into your work?
Oh, I would start with, what environmental variable would you want to know? That, okay, maybe there's other ways to get it, but it's super time consuming, or this, that, the other thing. Start there. Like, what are we trying to, if it's like temperature, you don't need microbes to tell you. Like, temperature is relatively easy to measure, like almost all systems, right? But something like oxygen is really hard because it's so temporally variable, spatially variable. Oxygen can be hard to measure at relevant scales and so forth. But so I'd start with like what variables you want to measure, and then two, is there any reason to expect there might be some sort of microbial bioindicator? Because sometimes there might not be. But the problem is like, again, to go back to this prediction theme, is you need a lot of data to develop the models to say we can use this as a useful bioindicator. And this is true whether you're looking at algal bioindicators, animal, plant bioindicators, and then you need to test that on an independent data set and show that it works. So yeah, it needs to meet those three, like, is there a variable that you want to measure? Is there a reason that microbes could potentially be useful as a bioindicator? And three, do you actually have the data that you could use to show that it's useful as a bioindicator? Yeah.
This kind of goes back to the scale problem, but if you use microbes as a bioindicator for like historical data in any way, like if you were just trying to figure out like a certain event for like a wildfire, for instance, I'm assuming it would kind of change, like your microbial makeup and your landscape for your ecology, and like fire usually where the fire, didn't impact it, you could like compare and contrast there, use that as a bioindicator? Do you
Oh,
think?
for sure. Yeah.
Or
I
does
mean,
that become that question mark, it's just way too complicated and.
oh no, for sure. Or you could use so we know like after high intensity fire events, it tends to select for certain bacteria, at least at the very surface of soil, right? So maybe you could use those as indicators, or like more simply, look for charcoal and so forth. But as an example of that, so I got involved in this ancient genomics thing where they're like taking sediment samples that go back 100,000 years from permafrost soil or from, the epicenter of rice growing in China to try to reconstruct like what the ancestral domesticated rice looked like. So most of the data they get is microbial, bacterial actually. So they ask that same question, can we use all that bycatch? They call it bycatch, I don't, of this bacterial data that's preserved over tens or hundreds of thousands of years to tell us something about that environment, which is this exact same idea. So we've already been using this oxygen approach to say like, oh, was that paddy field inundated and anoxic 50,000 years ago based on looking at these ratios of genes? Like a more simple way you can do it is look at like, when did humans move into that system? Well, you look for human fecal bacteria that we know are indicative of human feces. And if you see that in the layer, you say, okay, we started to get sewage entering this system at that point in time. Yeah, and of course, you can do this with plant and animal DNA. It's just there's so much bacterial DNA often in these systems and so much diversity that you can play with and you can identify those bioindicators.
Well, one last question that I'll ask myself. Trying to connect this to yesterday's talk. Today you mentioned the complication of the relic DNAs.
Yeah.
Yesterday you showed a result where there was in a sample with high spatial variability, there was surprisingly low temporal variability.
Right.
Is that the low temporal turnover potentially an artifact of this?
Yeah, so I didn't mention it, but in that study of the temporal variation, we removed the relic DNA for that exact reason, because we worried about, well, we wouldn't see any temporal, it's just all this dead DNA. So we removed that relic DNA prior to the downstream analyses.
How do you know that?
It's because there's a dipropidium monoazide that won't enter intact cells, but it will bind up any DNA that's extracellular or in a dead cell. So you can remove that fraction. We often don't do it because it's a pain in the ass to do, but there is a way to remove that relic DNA signal. But again, this question of how old that DNA is, I don't know.
Let's thank Noah.
Thank you.
That's fun.
I don't know if that meant you. I enjoyed it. You're not the target.
Check the box for me.
Generated the ITS of Brenton State. Do you have your RQP snap?
I forgot to move.
Yeah, for the air thing.
Hey, here it is.
Yeah, that was sequencing.